Episode 8: The Cambrian Explosion, Burst of Animal Diversity in Oceans. Part 1: The World Before the Explosion. Introduction: A planet poised for change.
541 million years ago, Earth stood at a turning point. For billions of years, the planet had teemed only with microbes, algae, and soft-bodied organisms whose delicate forms left little trace in stone. Then, over an interval short by geological standards, the oceans filled with shells, limbs, eyes, and jaws. Almost every major animal design that would ever exist appeared for the first time. To later observers, this transformation would seem sudden, a literal explosion of life, but it was built upon long preparation. The Cambrian world inherited the chemical and biological legacies of the Ediacaran: oxygenated seas, nutrient-rich shelves, and genetic toolkits already primed for complexity. The Cambrian explosion unfolded between 541 and 515 million years ago, an interval of roughly 25 million years in which taxonomic diversity increased tenfold and morphological disparity approached modern levels. The event marks the formal beginning of the Phanerozoic Eon, the time of visible life. Exceptional fossil sites across every continent preserve both soft and hard tissues, allowing this period to be reconstructed with unusual precision.
The Earth at the turn of the Eon.
The early Cambrian Earth was warm and restless. The supercontinent Rodinia had fragmented into smaller landmasses: Laurentia, Baltica, Siberia, and Gondwana, surrounded by broad, shallow seas. These continental shelves were fertile nurseries, bathed in sunlight and stirred by tides that kept nutrients in motion. Atmospheric oxygen, rising since the late Proterozoic, hovered near modern surface values. Calcium and phosphate, washed from weathering rocks, enriched the oceans and tipped their chemistry toward mineral saturation. At the start of the Cambrian, the chemistry of ancient rocks shows a major shift in Earth's carbon cycle. More organic carbon was being buried in sediments, which in turn meant more oxygen was being produced and left in the atmosphere and oceans. Other chemical clues, like unusually high levels of molybdenum, indicate that even the deep ocean was starting to fill with oxygen. Measurements of iron minerals back this up. They show that oxygen-rich conditions were spreading widely. Together, these changes created an environment where animals that needed lots of energy, and therefore lots of oxygen, could finally flourish. Reefal carbonates built by archaeocyathid sponges appear by 525 million years. These organisms required both clear, oxygenated water and stable carbonate saturation. Their framework reefs, found today in Siberia and Antarctica, are among the earliest large ecosystems constructed by animals and provide tangible evidence of a newly oxygen-rich biosphere.
The Cambrian Substrate Revolution.
Before animals learned to burrow, the seafloor was bound by dense microbial mats. Ediacaran life clung to or rested upon these surfaces, but rarely disturbed them. At the dawn of the Cambrian, small worm-like creatures began to dig. Their movement mixed the sediment, carried oxygen deeper into the mud, and destroyed the old mat communities. This behavioral shift, the Cambrian Substrate Revolution, transformed marine ecology forever. The appearance of the sinuous trace fossil Treptichnus pedum defines the official base of the Cambrian in the global stratigraphic scale. These complex, branching burrows record three-dimensional exploration of sediment by bilaterian animals capable of coordinated movement and sensory feedback. Sediment redox profiles changed dramatically. Oxygen penetration increased from less than a millimeter in the Ediacaran to several centimeters by 530 million years, expanding habitable microenvironments and creating niches for detritivores and predators alike. This new churning of the seabed altered global geochemistry. Organic matter buried in anoxic muds was now exposed to oxygen, accelerating decay and nutrient recycling. Phosphate regenerated into the water column, fueling algal blooms, while carbon dioxide release stabilized the carbon cycle. For the first time, animal behavior itself became a planetary force.
The invisible preparation.
Although the Cambrian explosion seems abrupt, its genetic and biochemical groundwork had been laid long before. During the late Proterozoic, multicellular organisms had already evolved the fundamental developmental machinery, cell adhesion, signaling pathways, and pattern gene expression that would later shape animals. What changed was the environment's ability to support them. Rising oxygen allowed these dormant potentials to express themselves as anatomy. Molecular clock analyses of conserved genes place the divergence of major animal lineages, sponges, cnidarians, and bilaterians, between 650 and 600 million years ago, well before their fossil appearance. The explosion therefore represents not the birth of animals but their ecological release, when physical and chemical thresholds finally permitted large, mobile, energy-demanding forms to persist. In parallel, oceanic calcium concentrations climbed to roughly 25 millimoles per liter, double the Proterozoic norm. This shift destabilized organic membranes and favored biomineralization as a regulatory mechanism for excess ions. What began as internal detoxification would soon produce shells, skeletons, and reefs.
The first skeletal organisms.
The earliest tangible relics of this new world are the small shelly fossils scattered through lower Cambrian strata, tiny tubes, scales, and spicules composed of calcium carbonate or phosphate. They are the first widespread evidence of animals constructing durable skeletons. Some belong to primitive mollusks and worms. Others, like Cloudina and Anaborites, built nested cones that foreshadowed corals and annelids. Thin-section microscopy reveals growth lines and microgranular fabrics identical to modern biologically controlled minerals, proving that these were true biominerals rather than chemical precipitates. Oxygen isotope ratios within the shells yield seawater temperatures of 30 to 35 degrees Celsius, consistent with warm, equatorial shallow seas that promoted rapid carbonate deposition. The ecological consequences were immediate. Hard parts provided protection and new surfaces for colonization, launching an evolutionary arms race between predators and prey. Mineral skeletons also increased sediment stability and carbonate production, feeding back into ocean chemistry and further stimulating reef growth.
A world transformed.
By 530 million years ago, the quiet microbial seas of the Proterozoic had become vibrant ecosystems filled with movement and form. Burrowers re-engineered the seafloor, shelled organisms built reefs, and oxygen-rich waters sustained the first food webs of macroscopic animals. The stage was set for the flowering of the Cambrian fauna, the appearance of eyes, claws, and intricate bodies recorded in places like Chengjiang and the Burgess Shale.
Part 2. The explosion unfolds. Eyes, predators, and the Burgess Shale world. The opening of the Cambrian seas.
Within only a few million years of the first shelly fragments, the marine world became unrecognizable. Burrowing and skeletons had re-engineered the substrate. Now movement and predation reshaped the water column itself. Organisms that once drifted passively learned to crawl, swim, and hunt. The Cambrian oceans turned dynamic. Clouds of plankton drifted above reefs and sandbars. Worm-like creatures plowed through the mud, and new predators patrolled the depths. For the first time in Earth's history, life possessed eyes, armor, and strategy. Radiometric dating places the main pulse of diversification between 530 and 515 million years ago. Within this span, the fossil record of every major marine phylum appears: arthropods, mollusks, annelids, brachiopods, echinoderms, and chordates. Measured in evolutionary tempo, morphological disparity increased faster during this interval than at any later time in the history of animals.
The birth of predation.
Predation, more than any other ecological force, drove the Cambrian transformation. Once some animals began to consume others, every lineage faced new selective pressures. Defensive skeletons, rapid motion, sensory organs, and complex behaviors evolved in quick succession. The simple microbial mats of the Ediacaran could not have withstood such an assault. They vanished, replaced by living sediments filled with predators and prey. The appearance of boring traces in early Cambrian shells, tiny drill holes less than a millimeter across, marks the first direct evidence of predation. The shells of Cloudina from Namibia and Oman show circular perforations made by viewer-like mouthparts. These fossils predate the main explosion by nearly 10 million years. revealing that predation began locally before spreading globally. Experiments with modern marine ecosystems show that a single introduction of a new predator can increase prey diversity twofold through the creation of refuges and adaptive specialization. In the Cambrian, this ecological feedback occurred across entire oceans, amplifying diversity on a planetary scale.
The evolution of vision.
To hunt or flee required perception. The earliest eyes appear among Cambrian arthropods, compound organs composed of hundreds of lenses capable of detecting movement and shape. Trilobites, which dominated the seas for the next 270 million years, carried calcite-reinforced eyes whose optical precision rivaled modern insects. With sight came the birth of complex behavior, pursuit, evasion, and recognition. The earliest known hard, mineral-based eyes appear in a trilobite called Olanellus, about 521 million years ago. Each eye lens was made from a single, perfectly aligned crystal of calcite, a natural mineral, arranged so that light could pass through cleanly. This is one of the earliest examples of evolution turning a rock-like material into a working optical device. Similar eye designs are found in the fierce predator Anomalocaris from the Chengjiang and Burgess shale fossils. Its stalked eyes held up to 16,000 tiny lenses, giving it vision on the level of a modern dragonfly. The emergence of vision altered evolutionary dynamics. Predators could target moving shapes. Prey could detect approaching danger. This visual arms race explains the simultaneous proliferation of spines, camouflage, and rapid locomotion seen in Cambrian fossils. Evolution accelerated through the feedback of seeing and being seen.
The rise of the arthropods.
Among the most successful Cambrian animals were the arthropods, creatures encased in jointed exoskeletons that combined protection with mobility. From their ranks descended trilobites, crustaceans, and insects, the dominant forms of later eras. In the Cambrian, they occupied every niche: swimmers, burrowers, scavengers, and hunters. The Burgess Shale preserves incredibly detailed fossils of early arthropods like Marella, Yohoia, and Wapsha. Their well-developed gills, complex eyes, and jointed legs show that the basic arthropod body design was already advanced by 508 million years ago. Chemical studies of the fossils even reveal traces of chitin and proteins, the same materials found in the hard exoskeletons of modern insects and crustaceans. DNA-based molecular clock studies place the split between major arthropod groups, such as crustaceans and spiders, at around 540 to 530 million years ago, which fits perfectly with what the fossils show. Trilobites first appeared about 521 million years ago and quickly evolved into hundreds of different species. Because they spread all over the world and changed rapidly through time, geologists use them as index fossils to help date ancient rocks. Huge numbers of their shed exoskeletons have been preserved, showing that trilobites regularly molted as they grew. The patterns of these molts suggest they may have had complex life cycles and even gathered in groups to shed their shells, possibly timed with the tides or the phases of the moon.
The Burgess Shale and Chengjiang faunas.
The fullest view of the Cambrian world comes from two extraordinary fossil sites: the Chengjiang biota of Yunnan, China, dated 518 million years ago, and the Burgess Shale of British Columbia, 508 million years ago. Both preserve soft tissues in astonishing detail: eyes, gills, intestines, even nerve cords, allowing reconstruction of entire ecosystems. The Chengjiang Shale contains over 250 species representing at least 16 animal phyla. Among them are the earliest known chordates, hycoichthyes, and Mylocunmingia, which possess notochords and segmented muscles, the first vertebrate relatives. Pyritized gut tracts reveal predatory diets, while delicate fronds of early algae show the base of the food chain. The Burgess Shale, deposited in a deep shelf basin below a carbonate reef, owes its preservation to rapid burial by underwater mudslides in oxygen-poor waters. Chemical analyses show that fine aluminosilicate films replaced soft tissues before decay, capturing even cellular detail. Species such as Opobinia, with 5 eyes and a grasping trunk, or Anomalocaris, a meter-long apex predator, illustrate how experimental and unbounded Cambrian design could be.
The dawn of the chordates.
Amid the riot of arthropods and worms appeared a lineage that would eventually produce fish, amphibians, reptiles, and mammals. The earliest chordates were small, translucent swimmers with stiffened dorsal rods and gill slits for filter feeding. Their emergence marks the beginning of vertebrate history. Hycoichthyes ercaicunensis from Chengjiang, only 2.5 centimeters long, shows segmented myomeres, a notochord, and paired eyes, definitive chordate characters. Geochemical mapping of phosphorus confirms that its internal skeleton was non-mineralized, composed of collagen rather than bone. Slightly younger forms such as Pichia gracilans from the Burgess Shale display more advanced musculature, indicating early experimentation with undulatory swimming. The origin of the vertebrate head, a concentration of sensory organs and a central nervous system, is traced through these fossils. Gene expression patterns in modern fish embryos mirror the segmented structures seen in Hycoichthyes, linking developmental genetics directly to Cambrian anatomy.
The structure of Cambrian ecosystems.
By the height of the explosion, the sea supported complex food webs. Photosynthetic plankton formed the base, grazed by filter-feeding sponges, brachiopods, and early mollusks. Worms and trilobites scavenged detritus, while cephalopod-like predators and anomalocaridids ruled above. For the first time, energy flowed through multiple trophic levels, recycling nutrients and supporting large populations. Chemical signatures in Cambrian rocks show that the food chain of the time was already organized much like today's oceans. The nitrogen values increased steadily from one step of the food web to the next, revealing a clear hierarchy of producers, consumers, and top predators. Studies of the Burgess Shale also show layered communities, animals burrowing in the mud, others living on the seafloor, and swimmers above them. This kind of structured, multi-level ecosystem had never existed before in Earth's history.
Transition summary.
By the close of the early Cambrian, life had achieved complexity and interaction undreamed of in the Proterozoic seas. The invention of sight and predation created evolutionary feedbacks that filled the oceans with diversity. Hard skeletons fossilized this abundance, ensuring that from this point onward, the history of life would be written indelibly in stone.
Part 3. The architecture of animal life. The blueprint of bodies.
The Cambrian seas were filled with new shapes: coiled shells, jointed limbs, segmented trunks, and radiating tentacles. Beneath this astonishing variety lay a shared biological grammar, a system of genetic instructions capable of building eyes, wings, and hearts from a single ancestral script. The discovery of this hidden unity has transformed our understanding of the explosion. What appeared as a riot of unrelated forms was, in truth, the unfolding of a deep developmental architecture already in place before the Cambrian dawn. The key to this architecture is the Hox gene cluster, a family of genes that organize the body along its head-to-tail axis. Each Hox gene encodes a transcription factor that activates specific developmental pathways in defined regions. The sequence of these genes on the chromosome mirrors the order of their expression in the embryo, an arrangement known as collinearity. Comparative genomics shows that all bilaterian animals, arthropods, worms, mollusks, and vertebrates share the same fundamental Hox pattern. The duplication and diversification of these genes between 700 and 600 million years ago created the molecular potential for every later body plan. Fossils of early animals like Pichia and the first segmented worms show that bodies with repeated segments had already evolved. Chemical traces from the same time indicate that these animals were becoming more active and energetic. Oxygen measurements in Cambrian rocks also point to warmer oceans and busier ecosystems. Together, these clues show how new genetic abilities and rising environmental energy worked hand in hand to drive early animal evolution.
Skeletons. Chemistry turned to architecture.
Mineral skeletons, first seen as tiny shelly fragments, soon evolved into elaborate frameworks. Different groups adopted distinct strategies. Some used calcium carbonate in its calcite or aragonite forms, others secreted calcium phosphate or silica. These biominerals not only protected the organism but also acted as scaffolds for muscle attachment and mechanical leverage, allowing larger and more powerful movement. The widespread appearance of hard shells and skeletons began when the oceans became rich enough in calcium carbonate for minerals to form easily. Models suggest this happened because more calcium and dissolved carbon washed into the sea as the ancient supercontinent Rodinia broke apart. Early animals with shells, such as anaborites and Hyalothellus, built them from a form of calcite rich in magnesium. As ocean chemistry shifted back and forth over time, later organisms switched to using aragonite instead. These changes reflect how ocean conditions shaped the kinds of skeletons early animals could make. Phosphatic skeletons, such as those of early brachiopods and small shelly fossils like Microna, indicate local upwelling of phosphate-rich waters. Silicon-based structures, including sponge spicules, record distinct oceanic provinces with high dissolved silica. These regional differences in seawater chemistry produce the patchwork of biomineral types visible in Cambrian sediments. The formation of skeletal reefs, archaeocyathid sponge frameworks and calcimicro buildups represents the first large-scale biological modification of Earth's crust. Their construction trapped sediment, altered carbonate saturation, and created microhabitats for countless smaller organisms. Reef growth rates, measured from vertical accretion in fossil sections, reach 20 meters per million years, comparable to modern coral systems.
The developmental explosion.
Behind the morphological bloom lay a genomic one. Analyses of gene families across modern animals reveal a burst of duplication events around the Proterozoic Cambrian boundary. These duplications created redundant copies free to specialize, giving rise to novel cell types, signaling molecules, and tissue structures. Multicellularity had provided the framework. Gene duplication filled it with diversity. Molecular clock studies of transcription factor superfamilies, including the homobox, basic helix, loop helix, and zinc finger groups, show peaks in duplication between 650 and 550 million years ago. This expansion correlates with the fossil evidence for increasing cell type complexity, from simple epithelia in Ediacaran organisms to true organs in Cambrian taxa. The emergence of immune genes, structural proteins, collagen, elastin, and neural channels all date from the same interval, forming the biological infrastructure of animal physiology. The neurobiological revolution of the Cambrian is reflected in trace fossils showing directed movement and predatory pursuit. Neuron-rich nerve cords are preserved as carbonaceous films in Fuxian Huia from Chengjiang, demonstrating that arthropods already possessed centralized brains with optic lobes. These fossils link neural complexity directly to ecological innovation.
Tiered ecosystems, life builds the landscape.
The Cambrian seafloor was no longer a flat microbial plane. For the first time, life occupied vertical space. Burrowers lived beneath the sediment, sessile filter feeders and grazers anchored to the surface. Swimmers and predators filled the water column above. This ecological tiering established the structure of marine communities that persists to this day. Fossil bedding plane analyses from Burgess Shale localities show tiered assemblages with three dominant levels. In faunal burrowers at 0 to 5 centimeters depth, benthic organisms fixed to the surface, and nectanic swimmers within the water column. Energy transfer between these tiers produced efficient recycling systems, reducing the accumulation of organic detritus. Bioturbation indices rise from 0 to 4 across the early Cambrian, quantifying this expansion of ecological depth. Changes in the global carbon cycle show how life was reorganizing the planet during the Cambrian. The chemistry of Cambrian limestone rocks rises and falls in a steady rhythm, reflecting repeated periods when large amounts of organic material were buried and later exposed again. These natural cycles helped keep the level of carbon dioxide in the atmosphere fairly stable, high enough to maintain a warm climate, but not so high that the planet overheated. In this way, early ecosystems began to help regulate Earth's temperature.
Cutaway, the geometry of evolutionary space.
Paleontologists describe the Cambrian expansion not merely as a rise in diversity but as a filling of morphospace, the abstract volume of all possible anatomical designs. Early in the period, new body plans appeared in almost every available region of this conceptual space: segmented worms, jointed limbs, radial forms, shells, spines, and soft-bodied swimmers. Later evolution would elaborate within these existing frameworks but rarely invent new ones. Quantitative analyses of morphological disparity using principal coordinate data from Cambrian and Ordovician faunas show that by 515 million years ago, total morphospace occupation exceeded 80% of the range achieved by the present day. This pattern indicates that the explosion's essence lay not in species count, but in the origination of fundamental body architectures, a single burst of innovation that defined the limits of animal design.
Stabilization after the burst.
By the Middle Cambrian, around 510 million years ago, the frenetic pace of innovation slowed. Ecosystems stabilized. Extinction and diversification reached equilibrium. Trilobites, brachiopods, mollusks, and echinoderms settled into enduring lineages. The evolutionary frontier shifted from invention to refinement. Natural selection honed the efficient forms that would dominate for hundreds of millions of years. Fossil assemblages from the Drummian stage, 504 million years ago, show community-level stability lasting millions of years, with turnover rates below 0.2% per 100,000 years, the earliest known ecological equilibrium. This marks the transition from the explosive to the consolidating phase of the Phanerozoic biosphere.
Transition summary.
The Cambrian world was not a chaos of monsters but a structured experiment in possibility. Genetic architecture, chemical opportunity, and ecological interaction converged to produce the living geometry of animals. The body plans forged in these seas, segmented limbs, exoskeletons, gills, and eyes, would persist, with modification, into every future epoch. In the next chapter of Earth's history, this new biological complexity would stabilize and expand beyond the shallow shelves, giving rise to enduring ecosystems and the first great extinctions.
Part 4. Stabilization and Legacy. From Cambrian seas to the Ordovician world. After the storm.
By roughly 505 million years ago, the explosive burst of new body forms began to slow. The once experimental Cambrian biosphere matured into stable ecosystems with defined trophic structures. Life had colonized every available marine niche, burrowing sediments, reef surfaces, pelagic waters, and the pattern of evolutionary radiation followed by stabilization had become established for the first time. The chaos of novelty gave way to the order of persistence. From this balance emerged the foundations of the modern marine world. Fossils from the Middle to Late Cambrian show an important shift in how life was evolving. The number of species kept increasing, but the variety of body shapes stopped expanding as quickly. Most of the major designs had already appeared. Measurements of these fossils show that by about 510 million years ago, animals were already filling more than 80% of the basic design space we see today. At the same time, extinctions became far less frequent than they had been earlier in the Cambrian. This drop in turnover marks the beginning of a more stable and balanced set of ecosystems.
The persistence of the trilobite world.
Among the enduring victors of this new equilibrium were the trilobites. Their chitinous exoskeletons, compound eyes, and flexible segments proved adaptable to nearly every marine habitat. Trilobite evolution through the later Cambrian reveals both conservatism and innovation. Conservative in their overall body plan, yet endlessly inventive in form and ornament. They became the visual emblem of the Paleozoic seas. Over 20,000 trilobite species are known, spanning 270 million years of history. Cambrian trilobites show rapid provincial diversification: Olanellids in Laurentia, redlichids in Gondwana, paradoxidids in Baltica, indicating the emergence of distinct continental faunal realms. Growth lines in their eyes and exoskeletons record tidal and seasonal rhythms, demonstrating not only biological sophistication but also that Earth's day length had already stabilized near 21.5 hours. Trilobites' molting behavior created mass accumulations of shed exoskeletons preserved as fossil beds. Such deposits serve as ancient census points of population dynamics. Geochemical analyses of associated shales reveal periodic anoxia in deeper settings, showing that even amid overall oxygenated seas, local redox gradients continued to shape communities.
Reefs, currents, and carbonate worlds.
Cambrian reefs, initially built by archaeocyaths and microbial consortia, expanded into immense carbonate platforms. Their framework structures not only provided habitats but also influenced ocean circulation and chemistry. By trapping sediments and modifying local hydrodynamics, they established feedbacks between biology and geology that would echo throughout the Phanerozoic. Archaeocyath reefs, built by ancient sponge-like organisms, were thriving between about 525 and 515 million years ago, but then they suddenly declined, probably because the oceans cooled and their chemistry changed. Microscopic studies of these reefs show that they were held together by thin layers of helpful microbes that grew alongside the sponges. Chemical patterns inside the reef rocks rise and fall in regular cycles, showing times when large amounts of carbon were buried by reef growth. Their collapse happened just as the oceans shifted from favoring one type of mineral for shells and skeletons to another, a change caused by shifts in seafloor activity deep within the Earth. The huge shallow water platforms of the Cambrian, covering more than 30 million square kilometers, played a major role in removing carbon dioxide from the atmosphere. As these vast seafloor areas slowly broke down and reacted with seawater, they helped lock carbon into rocks. Layered sediments built up steadily, showing that living organisms were constantly producing carbonate material that formed new seafloor. Over time, these enormous platforms hardened into limestone, which later became the core of many mountain ranges. In a very real sense, today's mountains are built on the remains of Cambrian reefs.
Extinctions and recoveries.
Stability was never absolute. During the Cambrian, life went through at least 3 major extinction events. Each one wiped out many species and opened up space for new kinds of organisms to evolve. The biggest of these extinctions happened around 485 million years ago, right at the transition into the Ordovician period. It wiped out many families of trilobites, the last of the archaeocyaths, and several early groups of echinoderms. But within just a few million years, the surviving species spread out and evolved into a wide variety of new forms. This burst of diversification kicked off the Ordovician radiation, the second great wave of increasing marine life on Earth. Around 517 million years ago, the Endbatomian extinction struck at the same time as major chemical shifts in the oceans and the spread of dark, organic-rich muds. These rocks show that oxygen levels collapsed in mid-depth waters. 2 later crises, the Endrumian and the Furongian events, between about 504 and 489 million years ago, show the same pattern, suggesting that low oxygen zones expanded again and again as the climate shifted and nutrients were recycled through the oceans. Together, these events show that by this time, life and Earth's chemistry were tightly linked. Changes in productivity, oxygen levels, and extinctions were now connected parts of the same system. Recovery after these crises saw the rise of new groups: articulate brachiopods, cephalopods, and the first planktonic graptolites, which would dominate Ordovician ecosystems. The continuity of lineage across such disturbances demonstrates the resilience of the new Cambrian-style food webs.
From Cambrian balance to Ordovician expansion.
As the Cambrian gave way to the Ordovician, the biosphere expanded both in diversity and in depth. Coral-like organisms replaced the extinct archaeocyaths as reef builders. Brachiopods and mollusks spread into cooler, deeper waters. Plankton diversified, transforming ocean chemistry and enhancing carbon sequestration. The slow, steady evolution of these systems turned the episodic Cambrian burst into a sustained geological revolution, the long-term oxygenation and biological modernization of the planet. Cutaway, chemical clues in ancient rocks show that by about 480 million years ago, the deep ocean had gained enough oxygen to stay permanently oxygenated, ending the long history of dark, oxygen-poor deep waters. This change made it possible for more complex food webs to develop in the open ocean and allowed seafloor animals to live at greater depths than ever before. What followed was the Ordovician radiation, a long surge of evolution that lasted until about 445 million years ago and ultimately tripled global biodiversity compared with the Cambrian. As plankton communities became more complex, they grew better at carrying organic material from the surface ocean down into the deep sea. Layers of early Ordovician sediment show repeated episodes when large amounts of this material were buried on the seafloor. Each of these events removed carbon from the atmosphere and helped cool the climate. Together, they show that the changes set in motion during the Cambrian had begun to let life itself influence Earth's temperature, creating a feedback system between biology and climate that still operates today.
The legacy of the Cambrian.
Every subsequent chapter of biological history traces its lineage to the Cambrian. The genetic blueprints written during that explosion have never been erased. All animals, from insects to mammals, bear the imprint of that first experiment in complexity. The trilobite's eye and the vertebrate skull share the same developmental logic. The coral reef and the Cambrian spongebank operate by identical ecological rules. In the Cambrian, evolution established its permanent vocabulary. Studies of how different animals develop show that the genetic systems used to build limbs in insects, vertebrates, and segmented worms all come from the same ancient ancestor. Evidence from both DNA and fossils places this ancestor right at the beginning of the Cambrian, around 530 million years ago, the time when evolution was at its most inventive. This means the Cambrian explosion wasn't just a burst of new body shapes. It was the moment when the basic genetic toolkit used by all animals today was first locked into place. Geologically, Cambrian sediments comprise some of the planet's richest archives of biogenic carbonate and phosphorite, the enduring record of biology transforming rock. Their isotopic cycles provide the first high-resolution calibration of Earth's biogeochemical rhythms, establishing a synchrony between life, ocean, and atmosphere that continues into the present.
Conclusion: the first age of animals.
The Cambrian explosion ended not with collapse but with integration. In its wake stood a world permanently altered, oxygenated, structured, and inhabited by creatures with eyes to see and skeletons to endure. For the first time, the planet's surface was shaped not only by chemistry and physics but by perception, locomotion, and behavior. The invisible biological foundation built through the Proterozoic had manifested in visible form, and the Earth had entered the age of animals, a world of interaction, predation, and evolutionary conversation that has never ceased. In the next episode, life leaves the shallows. Plants, fungi, and animals begin to colonize the land, and the blue world that birthed them will share its vitality with the continents above.